Cast.
The broadcast is now starting. All attendees are in listen-only mode.
Okay, guys, you're live. Where you go?
Okay, hello everybody, and thank you for joining us for this webinar today. My name is Andrew Forbes from Marsh Uni School of Public Health Preventive Medicine, and I've been the organiser of this session. I'll be hosting it today together with Madeline Enright from ACTA, who's been key in organising our participation in the session. Good morning. This session today is the first in a series of webinars organised by the ACTA Reference Group on Innovative Trial Designs that I chair jointly with Kate Lee from the Murdoch Children's Research Institute. So this series is called a Hackathon of Novel Trial Designs, but we don't actually know why that Hackathon title came about, because that's typically an IT-like venture. We get a whole lot of computer programs together trying to solve problems in a record amount of time. But the analogy with what we've got is we've got the clinical trials that we need to solve with novel trial designs, but we're not trying to solve them in a record amount of time, but we'll call it a hackathon nonetheless. So the idea of this series is to present novel clinical trial designs, either for trials that have already been completed with such designs, trials that are in progress, trials that are just in the planning or protocol stage, or even trials in which we've just got the seeds of ideas of how we might use some novel design for a particular trial. The main idea is to illustrate some trial designs that are smarter than the conventional standard two-arm parallel group design. So in today's session, we've got two speakers presenting their innovative or smart trial designs. Each speaker will speak for about 15 or 20 minutes, and we hope to have some good discussion after each presentation. Now, in your interface, there's it's called a chat box, actually, or question box or chat box in the lower right-hand corner. And so you can submit questions using the chat box at any time during the presentation. We'll collate them. Madeline and I will collate them, and then we'll read them out at the end of each presentation. We've also got an online sort of virtual panel of two people to lead off the discussion for each presentation, which we'll do first after the presentation, and then we'll follow that by the questions that are being submitted. If you're having any and the winner of the People's Choice Award of the four presenters that we're presenting will be, over our two sessions, will be invited to present their ideas at the ACTA conference in October this year in Sydney. So let's get going now with our first of our presentations. So our first speaker today is Professor David Paterson, who is the Director of the Centre for Clinical Research at the University of Queensland. He's also a consultant infectious disease physician, and his work has been recognised internationally as part of the Thomson Reuters highly cited list in multiple years, 2015, 16, 17. I assume David also 2018, trend going. His work research focuses on the molecular and clinical epidemiology of infections with antibiotic-resistant organisms, with the intent of translation of knowledge into optimal prevention and treatment of these infections. And he's now added novel clinical trial designs to his repertoire, and has recently conducted the world's largest trial on antibiotics for antibiotic-resistant bacterial infections. So it's fitting that our general theme of this series is about smarter clinical trial designs, and our first talk from David is actually about a smart design involving actually SMART being sequential multiple assignment randomized trials, and David's going to embed that within his talk about clinical trials to mimic clinical practice. And I'll hand over to you, David.
Fantastic. Thank you very much, Andrew. As Andrew mentioned, I'm an infectious diseases physician. I'm not a statistician, so please forgive my ignorance. And just as a little bit of background, I left Royal Brisbane Hospital and the University of Queensland in the late 1990s and went to Pittsburgh. And there I studied the molecular epidemiology of antibiotic-resistant bacteria. And that was all very interesting. And I remember calling my father once saying I'd discovered a new gene. In fact, that gene was the 131st slight variant of a previously discovered gene. But that got, at that stage, that got published, and I was all very excited. But interestingly, in Pittsburgh, I shared a bed with Ronaldo Bellomo. Now, that might sound a bit funny, but basically the house I lived in Pittsburgh was previously occupied by Ronaldo. There seemed to be a few generations of Australians. And obviously, therefore, we had this connection. And Ronaldo convinced me that, studying antibiotic resistance genes was all very interesting. But if you're a clinician, what you really want to do is study things that are going to change clinical practice. And so clearly, undertaking clinical trials is the way to do that. And so over the last 8 to 10 years or so since I've been back in Australia, that's really been my emphasis. So the trouble is that from an infectious diseases physician point of view, the trials that are done, and really the vast majority of randomized trials, are done for registrational purposes for the FDA or the EMA. And so it's a new antibiotic versus a control antibiotic. Always that control antibiotic has to be approved for that particular indication. And typically in these studies, they're syndrome focused, and there has to be very, very little prior antibiotic use for that patient, for that episode before enrollment. And so realistically, the vast majority of patients randomized into these trials actually come from Eastern Europe. And there, obviously, general practice and hospital emergency rooms are set up in such a way that a lot of people present quite late. They have not had preceding antibiotics from their GP, and therefore they're eligible for these sorts of trials. But really reflecting on that, it's not really consistent with how an infectious diseases physician would practice. And therefore the trials really have not a lot of relevance for day-to-day practice. One other thing that we note is that as long as an antibiotic has been FDA approved for that indication, it can be judged as the control arm, even if antibiotic resistance is such that there's a substantial amount of resistance to that antibiotic. So we're now seeing trials where the experimental arm, there may not be much resistance at all. The control arm, up to 40% of patients may have organisms that are resistant to the control antibiotic. And so it's not really a very fair or adequate comparison. So that's the sort of status quo. Just to sort of step back a little way, if we're talking about infectious diseases trials and bacterial infections, unfortunately, the current status is very much like it has been for the last 75 years. You come to hospital sick or you get a fever in hospital, cultures are taken, 24 hours later the laboratory can start to say what type of bacteria it is, and typically 48 to 72 hours after you've presented with the infection, we get susceptibility results back. And so that is sort of now indicative of two potential decision points that a clinician has to face. The first is the empiric antibiotic therapy choice. So that is on day 0, when that patient is sick, before we know the organism, before we know the susceptibility profile, a doctor has to make a decision about what antibiotic to choose. Then on day 2 or 3, when we know the organism, we know the susceptibility profile, there's another time at which a doctor needs to make a decision, and that is, what definitive antibiotic therapy should I choose for that patient? And that's going to be impacted upon by the organism name and also its susceptibility profile. So recently we've completed a large trial which was published in JAMA, which looked at that second decision point. Now, I'm not going to go into the ins and outs of the trial, other than to say that we were looking at 2 different antibiotics, and we were looking at which choice was the better choice for definitive therapy. So basically, we enrolled patients at 42 to 72 hours after they'd become sick, and we were interested in whether a particular choice, piperacillin tazobactam, was just as good as meropenem for that definitive antibiotic choice. And I have to say, a lot of our colleagues just couldn't understand this. They said, well, you haven't considered what was going on for empiric therapy. And I would say to them, I don't care. The trial was all about what would happen for a clinician, and this is something every week when I do an ICU round we're faced with. We've got a person who's now got in this situation, a ceftriaxone resistant E. coli or Klebsiella bloodstream infection. And I now know, because it's ceftriaxone resistant, I've got 2 choices, either piperacillin tazobactam or meropenem, and which choice is the better choice. And in fact, in this trial, we found that patients who got one of those choices, piperacillin tazobactam, actually had a higher mortality at 30 days. So just going over that again, this trial was all about the definitive therapy choice, and we didn't really care what the empiric therapy choice was. But there is an opportunity to study both of those decision points. In other words, randomize patients at that empiric therapy choice. Now, that's going to require a fair bit of work. We're going to have to work out ways in which we can get early consent, or maybe even pre-consent. For example, you know, a person comes into a hematology unit, they've become neutropenic, they're not yet sick with an infection, but at that stage, when we know there's a high likelihood they'll get sick, we may have to consent them at that stage and say, we're going to study that very, very early antibiotic choice on the first day of your illness. Because to be frank, it's tough to get consent from sick people when they're having fevers and rigors on the very initial day of their illness. But then we also have an opportunity, as we did in that previous trial, to randomize people into their definitive therapy. So I'll give you an example here. So the person has an infection that we were interested in studying on day 0. We could potentially randomize them, for example, to those 2 antibiotics we studied in a definitive sense, randomize people into their empiric therapy options. And then when we get back the results of their susceptibility testing, that's AST, antibiotic susceptibility testing. S means sensitive or susceptible, R means resistant. And also at that stage, which is now day 2 to 3 into their illness, we can determine whether they've had any significant adverse effects from their chosen therapy. You know, for example, if they develop renal toxicity or a significant rash, and that might also be an indication to change therapy. But then we have an opportunity to randomize patients. And here I've only indicated some of the potential randomization choices. And in this particular example, because we're seeing a lot of antibiotic resistance, I've only indicated that we may randomize patients to different options if that initial choice was the inappropriate one, there was resistance to it. But we could also randomize at a higher level to their definitive therapy option if there was susceptibility. So it is something of a complicated design, but it does very much reflect what happens in clinical practice. We have to make a decision on day 0. We have to make another decision on day 2 or 3 when we get susceptibility testing results back, and we get we have some knowledge about the tolerability of the antibiotic choice. Now, I won't go in, because of interest of time, into the different outcomes we could measure, but in conjunction with colleagues in the United States, we've looked at some ordinal ranking, desirability of different ordinal rankings of outcomes that would encompass both cure and toxicity and potentially also quality of life. But that's a whole topic in of itself. So this sort of concept has been coined by one of my colleagues, Scott Evans, who's a statistician at George Washington University in Washington, DC, as SMART compass. So sequential multiple assignment randomized trial. In other words, there are multiple opportunities to randomize a patient. And in this particular example, we're comparing personalized antibiotic strategies. So here, instead of just comparing antibiotic A versus antibiotic B, which is what a drug company would do, we're trying to compare decision-making strategies very much to mimic clinical practice. So an example, which is something we're faced with. So on day 0, do we throw the kitchen sink at the patient? Do we use our biggest gun antibiotic? Or do we start with a pretty aggressive approach, but one that we know is not going to cover all of the bases? And, you know, why, you might say, why don't we just give big guns to everybody, is that unfortunately, the broader the spectrum of antibiotics, the more collateral damage we may see, whether that be with toxicity or with development of antibiotic resistance. So that empiric therapy choice, there's a lot of tension at the moment between what we should do. Should we throw everything at the patient initially and potentially then ease off or de-escalate? Or if we've had a good response initially, should we just stay the course? And now with the advent of resistance, should we actually be studying salvage therapy? What happens when our initial choices are wrong and we actually have antibiotic resistance and have to modify our antibiotic choice? So I'll just illustrate the potential issues here where we may have actually 4 strategies that we're comparing. So the first strategy is really to compare big gun therapy at first, or middle of the road therapy, and then to compare a strategy where we keep going with the big gun therapy. So that might be strategy 1. We've started with it empirically. We now know the organism is susceptible to that very broad spectrum antibiotic, and we keep going with that choice. Now, also involved in that situation is no antibiotic choice is perfect, and we may have resistance, and we could potentially have an opportunity to compare at this point, even though I haven't illustrated it here, compare different salvage options. So this is potentially option 1, strategy 1. Go with your biggest and best and keep it going even after we've found that it's susceptible. Now, a second option may be we've started with the big gun therapy. Down here, we then randomize patients, as I've mentioned, to either staying with the big gun therapy or narrowing the therapy to the, I guess, most ecologically sound choice that is still going to kill the organism of interest. In other words, de-escalate from that big gun therapy. And in fact, you've noticed I'm not saying a particular antibiotic choice like meropenem here is the big gun therapy. It might depend where you are. If you're in New York City, your big gun therapy might be a new $1,000 a day antibiotic called ceftazidime-avibactam that's going to cover all of the bases there. If you're in Adelaide, big gun therapy might be piperacillin and tazobactam if there's not that much resistance. But we could potentially design a strategy trial where we're comparing strategies. We don't really even have to compare the particular antibiotic choice. It's really the strategy of going really broad, really early. And similarly, when I'm talking about the narrowest choice, that may very much vary according to the antibiotic susceptibility profile for that particular patient. You'll see a third choice here is if you're randomized to a middle of the road strategy, one that's pretty good, but it's not going to necessarily cover all of the bases, and we've chosen that because it might be ecologically more sound. And again, we have a randomization step there where we might continue that middle of the road strategy, or we might go down to a very narrow spectrum. And again, even though I haven't sort of illustrated it here, there would also be an opportunity to randomize our salvage options. And the fourth option, as I've mentioned, is that we could start middle of the road and then be randomized down to a narrow option. So these are the 4 strategies, and this is something that we're confronted with every day. When we do our ICU rounds, you know, what is the best thing? You know, there's a tendency, according to some prescriber personalities almost, to start with the big gun and stay the course with it if we can, whereas other people may be more conservative and maybe more inclined to consider the effects on the microbiome of every antibiotic choice, and might be, you know, more inclined to start with middle of the road choices and then try to narrow if at all possible. We don't know which of those courses, these strategies, is actually the best one. It's almost like we could do a vote compass. I don't know if those of you have seen ABC Vote Compass, because across prescribers, there is going to be a variety of strategies which is favoured by different individuals, but we really don't have an evidence base to back that up. So just to sort of summarize this smart compass idea, it could address several types of questions. The biggest one is, what is the optimal overall strategy? But we could evaluate empiric therapy strategies in isolation. We could evaluate definitive therapy strategies in isolation, including salvage therapy options. And we can explore more refined antibiotic tailoring choices. And we could potentially have as much efficiency as a traditional multi-arm study, because you could go back and say, well, why didn't you just have 4 different arms and just set up the study in that way. But we could potentially design it in such a way that individual patient data could actually contribute to informing multiple strategies. So potentially an advantage of this trial design is that it's pragmatic. It does mirror decision making that's done every day. It focuses on optimal strategies rather than individual drugs. And it's ethically attractive rather than the situation we're seeing now in some of the registration trials where there's no adjustment according to susceptibility pattern. Patients in Eastern Europe just get stuck on whatever they're randomized to for the duration of the study, and only at the end analysis are people saying, oh, look, in fact, 20 or 30 or 40% of the particular choices were actually inappropriate according to resistance. Now, disadvantages, obviously, there's a cultural shift that would be necessary. And even in our trial that was published in JAMA, we had a lot of people who just didn't get it, that we were only studying definitive therapy. It's logistically complex. There's the potential that you're going to have 2 stages of randomization. If we're going to do a number of different therapeutic adjustments, could you actually do this in a blinded way? Sample size calculations and analyses are potentially more complicated, and there may be a need for weighting of patients to obtain appropriate estimates of effects and associated standard errors. So in conclusion, what I'm trying to think about, and certainly my colleagues, such as Scott Evans, who I really want to salute for his inspiration in this regard, is to get away from how industry-sponsored trials are done. And in this era of antibiotic resistance, how are we going to study different strategies of antibiotic prescribing that are going to cover both the empiric and definitive choices? And one potential way, and I just want to sort of put it out there, this is just being tossed around at the present time. We don't yet have a study underway. How are we going to do this smart compass, this sequential multiple assignment randomized trial in this situation for comparing personalized antibiotic strategies? So thanks very much. I'll now get rid of my screen and open it up to questions.
Thanks very much, David. Very stimulating presentation. We've got two discussants set up to ask a few questions from there, but please encourage any of the audience to send in any questions you may have either now or we can also have towards the end of the session again. Our discussants are Julie Marsh from the Telethon Children's Research Institute. Sorry, I've messed that up, Julie, in terms of the title. And Stephane Heritier from the School of Public Health and Preventive Medicine at Monash Uni with me. So we might hand over to Julie first, or any questions or comments you might have.
Thank you, David. That was an excellent presentation and particularly relevant. We're doing a platform trial in cystic fibrosis. That's exactly what we're facing as well. There were certain aspects of it I particularly enjoyed around the idea of these trials becoming embedded in healthcare. So each centre, particular strengths around different sites being able to choose their own big guns or middle of the road therapy to reflect the different resistance patterns as well. I thought were major strengths of it. I suppose the issue you were skirting around was how you're going to analyse it.
Exactly, and I mean it could just be analysed in a standard frequentist way in which we're really comparing the 4 different strategies, A versus B versus C versus D. But it's intriguing, isn't it, to think, is there a way to integrate this into a Bayesian adaptive design? And I know, Julie, at your centre, that's something people are looking at, and so I'd really value comments as to whether there could be that it could be adapted into that manner of analysis.
Oh, absolutely. In fact, I'm thinking how we can incorporate some of exactly what you've specified here into how we're designing our trial. I quite like the idea of because we're worried about clinicians coming on board. There's very much that impression of, well, we've always done it this way. And I think overcoming one of the cultural aspects is by putting these different strategies into domains that, if you wish, the clinician can choose which domain they wish to be randomized within. So they have their a priori, this is the strategy I believe that works. You can randomize them just within a strategy and treat it as a domain and evaluate their patients within that domain and share them across the other ones to come up with some really interesting, I mean, it would have to go down the Bayesian route really for the borrowing that goes on, so you could work those out. But I have to say, we cheated and went the opposite way and said, we're interested in, you have to make a decision on day one. And we focus more on the empiric antibiotic choice, because that was when you're completely blind, you know, where should we even start? Bearing in mind that many cystic fibrosis patients could be delayed up to a week or 2 before they're even admitted to hospital for treatment.
Yep, interesting.
But absolutely, come and talk to us.
I will. Thank you very much, Julie. Great.
And just cutting here, it's Andrew in here. I think there's a lovely synergy between your design for this talk and what we'll hear from John Symes as well, particularly in light of Julie's comments about sort of extending this to a sort of a broader adaptive sort of platform setting. Okay, Stephane, do you have some comments you'd like to raise? Oh, I think you're still on mute, Stephane. We still can't hear you, Stephane.
Stephane, you're no longer on mute. You should be able to talk now.
Now?
Yes, it's better. Yep.
Maybe that was the earplugs, I don't know, so I removed my earplugs, I don't need them anyway. So first, thanks David for this very interesting presentation. It makes a lot of sense to design your trial as a smart trial, because it's what it mimics what you are doing in practice, and it's not so easy to do. As far as I know, it's the first I've seen presented in Australia. The idea dates back to the first time it was introduced, I think, in 2009. So it takes time. Susan Murphy introduced that. She struggled to get this idea across, but it's very good to see that you have been able to do it. I really like the design. So now I have a few questions for you. One is about the sample size calculation. You mentioned that a little bit. I guess, how do you go about it to get your sample size in such a complex trial? You need to simulate, you need to identify upfront the strategy you want to test, and that may depend on, sample size may depend on that. How do you go about it?
Yeah, so I mean, it's unfortunately something we have not yet got to, but clearly we would need to evaluate a number of different variables. One is the extent of resistance in different trial sites, because as I sort of alluded to, you know, if you're in an area where there's a lot of antibiotic resistance and therefore there are more people going to potentially need salvage therapy than in sites where there's not a lot of resistance, you know, that's going to be an important variable to model. So I'll have to sort of plead ignorance in terms of, you know, what are each of the explicit steps that we would need to undertake to adequately determine a sample size. But I would love to hear from others who would have some ideas about exactly how that might be undertaken. Well, Stephane, do you have some thoughts already?
Well, I haven't really thought too much about this, but I suppose you could simulate this, but you would need to have some ideas of what's going to happen, which might be a little bit difficult. So I don't know. There might be some ways of finding formulas. If you go Bayesian, like we discussed before with Julie, and you are going to simulate anyway, this is probably this is the only way to get it done. So I don't have a clear view now on this. I'm just I was a bit curious about it. Then also this question about, in the end, you are going to test several strategies, right? Or I suppose you define the strategies you want to test. It may be more than one. Do you sort of take into account the multiple testing you're going to do, or you leave it standard technique, standard way of standard procedures, adjustment, or some sort of adjustment, I guess?
Yeah, again, that's a very good point, and I want to sort of say again, I'm not a statistician, so I'll have to ask Scott Evans what his plans would be in that regard. Again,
right.
Stephane, your off the cuff sort of thoughts about what might be the way to do this?
Well, I don't really know. Maybe you don't necessarily need to adjust. Depends on what you want to really see. So, well, let's ask maybe a more practical question. When you presented, you talk about the treatments like being big guns and what was it, the other one? Middle of the range. So, and you said that big guns might not be necessary. I mean, if I understand well, might not be exactly the same depending on the site and so on. So, I mean, if they are too different, for me, it's a little bit of a problem. It seems test a strategy, but if the big guns are quite different, I mean, do you allow that, or you have somehow some sort of standardization of the big guns or whatever the treatment?
I think there would be an opportunity to standardize it according to the proportion of resistance at any particular site. So, for example, you know, at sites where fewer than 5% of patients may have resistance, there might be that even may be a different strata, or it may be a different, certainly it'd be a different big gun option compared to sites which may have, for example, 15 to 20% resistance. So I think you're right that when we might have different drugs within the same strategy, there could be, I guess, criticism that there might be different effects of each individual drug. In a way, we don't really care because we're just looking at the strategy overall, and the concept of starting really broad early, so that it could be generalizable to as much of clinical practice as possible. But you're right, it is an obvious criticism that if you actually have different drug choices within your strategy, you may have potentially different effect sizes. So it's again an interesting thing to consider.
Yeah, because in the end, you're going to write your conclusion. It is close to the end. You've got some effect of this particular strategy. You are going to say this strategy is better, but in the strategy, you've got different treatments. It's kind of, if they are too dissimilar, that may be a problem. If they are sort of similar, I don't have any issue with that. You want to test the strategies quite more or less the same. But I don't know. I mean, I haven't, just from ideas, I was thinking while you were talking.
I think in antibiotic trials, the response rate in susceptible, if the pathogen is susceptible, are fairly similar. I mean, right since the 1990s, it's all been done on non-inferiority base, or we used to call it equivalence. I think it's more trendier to call it non-inferiority nowadays. So I think it's a slightly different issue when you work in anti-infectives. I mean, I know things like the new hepatitis C treatments, they're all up at 90% cure rates. So the problems really kick in around toxicity and resistance patterns.
Yes.
And with the multiple testing, of course, if you're Bayesian, it's less of an issue.
Okay, can I jump in now? We've got an audience question here. This is from Dan Kent. So David, do you foresee any issues with designing consent forms which allow for the movement or crossover of patients between different arms?
Yeah, so this is a piece of work that's being undertaken at the moment. What is the acceptability of patients, or maybe we might call them consumers? What is also the acceptability amongst ethics committees of this sort of pre-consent or early consent? Because it is a major issue for us in antibiotic trials, that if we're going to study strategies that are going to include empiric therapy strategies on day 0, we are going to run into difficulties getting enough people to give consent or get proxies, etc., particularly if we've got a very short time frame to enroll patients. Now, obviously, some of those challenges have been overcome in critical care or emergency medicine trials, but it is important for us to get feelings of emergency, of ethics committees and also patients about the acceptability of doing this early, and potentially, giving consent that you might end up having another stage of randomization as well. So the concept would be that you give consent to the whole package, not each individual part of the package.
Oh, thanks very much, David. Sorry, I was just on mute there. If anyone has any questions, please feel free to write them in. We can come back to that at the end. But thank you very much, David, for your presentation. There may be coming at the end. So we might, and thank you to Stephane and Julie for your comments as well. So we might now move on to our second presentation today. And this is from Professor John Symes from the NHMRC Clinical Trials Centre in Sydney. Now, John's been a key leader in national and international clinical trials development for many years. He's the founding director of the NHMRC Clinical Trials Centre, He's also director of the Sydney Catalyst Translational Cancer Research Centre. He's the founder of the Australian New Zealand Clinical Trials Registry and also co-founder of ACTA, Australian Clinical Trials Alliance. So obviously we owe a lot to John for his tireless work in establishing these critical pieces of infrastructure for clinical trials nationally. So John's going to sort of follow on nicely from what David talked about with standard of care randomised trials to enhance the science of medicine. So I'll hand over to you, John, now. Thank you.
Thanks very much, Andrew. I'll just see if I can get my slides working. And if someone could just confirm, I'll share my screen. So could you somebody just confirm that they're there so people can see them?
Yes, John, they're fine.
Excellent. Okay. So look, what I'm going to talk about is a fairly radical idea about how we could try to make randomization a much more routine part of delivery of standard of care. Coined the name SOCRATES of standard of care randomized trials, enhancing the science of medicine. And this approach is to try to look at whether we can have whenever we have more than one option of standard of care that's being considered for a patient, is there a very simple mechanism to be able to randomize that and address therefore a whole range of questions which would be extending the evidence base from more well-defined patient populations to real-world patients, a little bit along the same motivation as our previous talk. The process for this is to keep the studies very simple and to look at this as a mechanism of both establishing a database of randomized evidence through a web-based simple collection device, but also, I'll come back to this at the end, consider using the randomization as a way of adding to what's currently talked about in terms of big data, where we're looking at observational data usually to inform decisions about clinical practice, etc., but usually where randomization is not part of that. So could this actually become an additional important variable in such databases? Now, this is this next part is really quite straightforward in terms of being able to, for this audience, to say why randomization is important. But I thought it's still useful. This is going to be still fairly key as to why can't we get away without randomization in other situations. So we know that large randomized trials are necessary to reliably demonstrate moderate treatment effects. There's a suggestion that if we've got better targeted therapies and the better use of information from genomics and other things, we can possibly get away without randomized studies. And in the future, if we are using big data with artificial intelligence and other things, this may be seen as an approach to avoid randomization, but I would contend that that will still be seen as suboptimal. Here's an example of a review of some of the older trials done in cancer over some 50 years, where new therapies were compared with the old therapies in randomized trials, where the hope based on the observational data, this would lead to improvements in outcomes. And for these particular studies, the average treatment effect was 0, i.e. the new treatment on average was equal to the old treatment effect. Sometimes the new treatment effect was significantly better, sometimes it was significantly worse. But if we had relied upon the observational data, for these studies at least, we would have made no advances in the treatments. Because of the randomization, the treatments over this side, which showed significant advances, significant benefits, are now would be adopted in practice, and the ones over here would not have been used. So through the randomization, we're able to make true advances in healthcare. Has that situation changed with novel therapies? And this is looking at novel biologic anticancer therapies. And there has, you'll see here, been a shift to the left of these curves. So on average, the trials show a slight advantage of the new therapy over the old one, but still with a considerable overlap. So quite a lot of uncertainty where if we simply adopted those targeted therapies without the randomised evidence, often we still would have been wrong. We're getting better for some of those, and so there may be still a further shift to the left. But I think the bottom line is that even with a whole lot of additional information, randomisation is still key to be able to ensure that we're making true advances in healthcare. So the idea of Socrates then is that somebody for many years ago, a great philosopher, had already said, but way back then, true wisdom comes to each of us when we realise how little we understand about life, ourselves and the world around us. And the philosophy, therefore, of using Socrates for this is that we need to continue to challenge both accepted dogmas and new dogmas which aren't based on reliable evidence. Just to go over a little bit more of the background or the rationale for this approach, a lot of treatments that we use today are still not based on reliable evidence, and therefore there's a need to still look at practical ways of increasing the randomised evidence base to guide future therapies. And there are several interventions where we do have good evidence, but they're not widely used in clinical practice, or not as much as we think they should be, either because the evidence is currently not applied, or because people consider the evidence is not applicable. The patients studied in the trials are not, the patients that we're faced with in front of us may not actually have qualified for those trials on the basis of eligibility, or they may also be in other settings. So there's a need both to apply the current evidence better, but also to look at systems for generating new evidence when we think that that evidence is not applicable to our patients. There are significant barriers in undertaking randomised trials in this setting, and so a lot of the comparative effectiveness studies, many do involve randomisation, but many also are based on observational data. And those barriers relate to the costs. They're not studies that are well funded, for example, as a new drug therapy, regulatory issues, ethics, and so forth. And so as a result of this, we make a number of decisions still based on the observational data. So what are the features of Socrates, and how might this be a fairly simple approach to try to have a major paradigm shift in this regard? It's based on 4 simple elements. So it's a very simple approach. Firstly, that you need to have 2 or more standard of care protocols for the patient and the doctor involved for their particular condition. The idea is that not only do you think this might be appropriate treatment, but in order to have ethics approval for this overall approach, it's a recognised standard of care based on a protocol, which might be a locally based protocol in a hospital, or a set of evidence-based guidelines or other things. Secondly, the doctor responsible for the care of the patient considers each standard of care approach appropriate without a clear preference for one over the other. Next, the patient also needs to be agreeable that receiving one of these options would be appropriate and without a clear preference. And finally, this model looks at having informed consent from each patient for the treatment options and the collection of de-identified clinical follow-up information. So that's all there is to it. But the question is, how do you actually then put that into practice? Well, first of all, the standard of care protocols or guidelines are ones which remain the responsibility of the hospital or the institution and the doctor caring for the particular patient. They're part of usual clinical practice, and so they don't actually have to appear in the clinical trial protocol. They'll be, the protocol, therefore, is a very generic one. I have a variation of that where one could still list specific references to such clinical protocols or standard of care protocols that might be eligible for studies, and I'll come back to that at the end in terms of how you might actually put this into practice. So the protocol, Socrates protocol itself, though, doesn't actually provide all the details of the treatments and the side effects and so forth. So what are some of the ethical issues related to this approach? Well, the process for getting consent and participation in the trial would be essentially the same as you would be doing if you were seeking consent for patient receiving their usual care. You'd be discussing what the options of treatment might be for their condition, what the current evidence for it was, whether that one or more approach might be appropriate, and then at the end of that, say if they're comfortable to have one or more than one of those approaches, rather than simply going ahead and saying, well, let's give this one, we'd say, we'll go online and we'll get particular treatment from the SOCRATES protocol. The patient would consent to that, and in addition give consent to use the identified data. There wouldn't be any additional tests or monitoring undertaken. This is providing usual care, and the information being used is analogous to a clinical audit of routine care. So the ethics for this is essentially therefore approval of the generic approach. One important aspect is that often clinical trials have a lot of information being collected in relation to adverse event reporting for new treatments, but this is essentially trying to mirror what happens for standard care. So if you had an adverse event, a suspected adverse drug reaction, which you currently can report to the TGA, for example, for a treatment that's being used in usual practice, the doctor would have that same responsibility within this trial, but there wouldn't be an additional responsibility to report that information back to the people running the trial. You could be collecting some of that information if you elected to, but you wouldn't require to have real-time reporting of adverse events, etc. For the study, the consent form, and we have a consent form has been put together for this, but I haven't shared it in these slides, would actually be, in a generic way, list the general approaches. The actual patient's medical condition and the post-treatment options would be added to the form by the medical practitioner, and then the side effects, the treatment goals, other options, etc., would be discussed with the patient in the same way as they would be for their usual care. But they wouldn't, that information would not be contained in the consent form. It would say that my doctor has explained to me all the potential side effects of these options. There'll be several other additional areas in terms of the consent form concerning issues about how the information is being used, privacy, confidentiality, etc. In terms of legal issues about do you need clinical trial insurance and other things, most of this is really the responsibility for the care of the patients remains with the patient's usual doctor and the institution where they're being looked after, and it's based on clinical protocols of standard of care, and therefore it would be covered by those same legal indemnity issues. There are two potential risks where the trial could be additional risks associated with the trial itself. One is that in collecting that information there were breaches in confidentiality. Here you would need to have information about the responsible clinician to be able to seek the follow-up, etc., but you would only have the de-identified information about the patient. And the second one, this is actually probably one of the major challenges, is that we're collecting information here about multiple treatments, multiple conditions without, I'll come back to the sample size issues as well, without a clear stopping rules of when we've got a sufficient evidence for a particular trial. And so if we were collecting this information but it never made its way in terms of future evidence-based guidelines, then it may be that we've demonstrated that a treatment's not effective, but we're not providing that back, and therefore the current guidelines were no longer relevant. So there has to be a mechanism to ensure the evidence we're collecting is made available in some reasonable manner to update the current evidence base. So how would the information be collected and updated? The proposal is to have a simple web-based centralised database with the primary clinician responsible for providing a simplified data record. At the time of randomisation, information would be collected on the patient's medical condition, disease descriptors, risk factors, potential risk factors that might relate to outcomes for that particular disease, some details of the particular protocols that were nominated, and then the treatments that were being given. And then the follow-up would contain just simply whether after completing their initial therapies, whether the patient had responded or not to therapy or progressed in terms of their disease, overall survival, a few other outcomes. We've got a version also looking at patient-reported outcomes, which would be something worth doing, but would also require then some additional logistics in terms of how that would happen. But the idea is to keep the information being collected at a very simplified level. The other aspect of this, which I'll mention now as well, is that if this information is being routinely collected and entered into the medical record, if the medical record is used in the future to look at databases of what happened to patients, you'll have an extra variable in there about a control therapy that the patient might have been, or an alternative therapy the patient could have been randomized to when you're interrogating those databases. So in terms of trial design and statistical considerations, this is quite radically different from what would happen in any standard trial, in that there's no predefined, standard trial or traditional trial would have a predefined sample size or a number of events in order to provide reliable evidence of a treatment effect. We'll also have monitoring of interim data to ensure that if there was clear evidence of benefit or harm prior to the trial completion, that information could be made available. In contrast, for this program, there'd be no pre-planned sample size for each question, nor any pre-planned interim analysis. Rather, you would need a mechanism to provide a regular update for each particular question, whether it might relate to a certain number of events. It could also be tailored to each particular question according to what other evidence was available. Now, the concept I'm outlining here sort of covers all medicine and all treatments, and of course that's going to be impossible. So you're going to start out by looking at subsets of that or various confined to certain disease categories, etc., rather than, or certain questions, rather than everything at once. But the end game, if we're thinking many years down the track, would be that this could be capturing everything. The data then would be provided in a publicly accessible database and also made available and that would be updated at predefined intervals, and it would also be made available to systematic reviewers, so it could be combined with other evidence from other studies addressing similar questions. There wouldn't be an independent safety and data monitoring committee for any single question, but you could have one overseeing the overall program to see that that sort of system was working. This is obviously very left field. It's out of the box. It's got lots of challenges with it, and that's why I'm hoping we'll have some great discussion in a moment about some of those. But I think one of the ways to think about this is what the world might look like in 20 years' time, 50 years' time, 100 years' time. If we're thinking here about not just how we traditionally do clinical trials, but what could be a world we could live in the future where we're making the best use of digital technologies, genomic technologies, a whole lot of things, and how, if we're thinking well in advance in that, then what could that future look like when a simple method to randomise standard care is an integral part of delivery of healthcare? Then I think this then has a lot of attractions. So firstly, big data will be used much more in the future to guide clinical practice and therapeutic decision making. My major concern about it at the moment is it is still all observational data. There are myriads of data points. We're going to end up drawing erroneous conclusions based on that, both in terms of multiple comparisons, but also because of the biased nature of the patients we'll have in those data sets in terms of how they were selected for therapies. So in addition to linking personalised treatment to genomic or molecular profiles, in addition to data mining using multiple sources for health records, I'm suggesting a third and critical component should be linkage of interventions to a randomised controlled therapy. So if, as a routine part of these future big databases, randomised controlled therapies is often included within them, then that's going to make those big databases so much more valuable. The second thing is to think about how we're currently using evidence in terms of routine clinical practice. We have, as Paul Glasio and others have outlined, there's a lot of wastage in the clinical evidence that's used, not being routinely used in practice. So think of a world in the future where we're doing some of this already, but doing it much more routinely, where one can go online, see what the evidence is, apply that evidence when it applies to our patient. But if for some reason that evidence doesn't apply to our patient, or we don't think it applies, but nevertheless that online system indicates that that particular treatment would be at least an option, and here's another option, that you could use that same database not just to implement a particular piece of evidence, but generate or extend the evidence base to new patients. So in the future, I'd like to think that there'll be many strategies for increasing randomized pragmatic trials. There's registry-based trials, of which I'm just talking about as an example, but which might be for specific clinical trials linked to a specific registry. There are issues of consent waiver, and the version I've given here doesn't contain consent waiver. Everybody for these studies is agreeable to consent to one of the options up front. But you clearly have to have models such as what we heard about before for the use of intensive care type studies up front. I've got listed another one there, the Torpedo 3060 study in preterm infants, where it's looking at 2 levels of study, what level of oxygen to start in terms of resuscitation. And that particular study already has approval for ethics approval not to give consent up front, but to have a consent waiver, and then go back and ask for consent subsequently collecting some information. But those not giving consent would still, not wishing to provide the extra information, would still be included in terms of their outcomes of survival and so forth as part of a, quotes, clinical audit. So there are other approaches in addition to the one I'm outlining here, but I'm just suggesting in the future we had a world where the routine use of randomization was significantly greater than it is at the moment for standard of care studies, mechanisms to achieve it, then I think this is one practical way of doing it. My final comment is we have currently put together a pilot protocol for SOCRATES. We're proposing with a patient information sheet and other information, which has been discussed with the chair of an ethics committee and with some in-principle support. We're looking at doing that in the context of some specific diseases. So one would be in lung cancer, where we're also doing some other information about defining what current evidence-based care is being used in practice. So as a proof of principle and just showing that some of these principles could be applied, we want to start small, demonstrate that it can be done. That particular pilot study will also collect information about just whether patients support the idea of this sort of approach. So I'll be asking information about the overall concept as well, before then trying to make it something that could be used more widely. So I'll stop there. I'm happy to take questions.
Thank you very much, John. Very stimulating talk. Got some panel discussants again. So Stephane Heritier, again, is one of the discussants. Also invite Julie Marsh, if she's still online, to have any comments, and I'll make some comments as well. I might just start off by saying that this is a radical design. I guess I'm already converted, that I think this is the design, randomized trial design of the future. And Stephane may be able to comment more about the REMAP-CAP community-acquired pneumonia platform trial, which shares a lot of similarities with this, where different patient subgroups get treated by different domains, and each centre may have a different set of interventions that they use. So I might hand over to Stephane, who's one of the chief statisticians on this trial, to talk about some of the similarities between what John's proposing and what's in REMAP-CAP.
Okay, Thanks, Andrew. I was not necessarily thinking about REMAP-CAP, but I was just trying to follow John's ideas. So, and for me, make the case of using more existing data, whether observational data or database or whatever is out there, or even big data, which makes a lot of sense. And if you want to use this to select standard of care, or you would like to use it to potentially identify treatments, is this both, or to come from, or the treatments to come from other sources?
So if we're talking about new treatments for which are not currently a standard of care, is that what you're saying, but might be reasonable options, that would fall outside the, I think is that what you're asking, that would fall outside the scope of what I'm talking about here. This would be, and one issue here is really, if you go to clinical standard care protocols, what information is currently listed as such a protocol? So sometimes are we already working outside that in terms of what people do in practice? So some of the challenges here will be, if you have, for example, if I take the lung cancer one I mentioned before, some standard of cares would be to give chemotherapy for lung cancer, and some people wouldn't get chemotherapy. And they wouldn't get it because their clinician would decide that because of comorbid illness or other things, there wasn't enough evidence to support it. They would regard that as a standard of care, but it wouldn't necessarily appear in any guidelines anywhere as a standard of care, no treatment. So one of the challenges we have here is, I think, some of the things in putting this into practice will be some of the standard of care options available be fairly limited. There are some natural ones, and I'll give you an example in a moment, for this particular trial, which is happening for coagulant therapy. But some of them will not easily fit into this. And so I think in parallel for what I'm proposing, we'll need to see the development of evidence-based guideline options. So not only this is a standard of care, but there's more than one standard of care. A lot of guidelines already talk about there's no defined treatment instead of following, but I think in parallel with this we'll need to see an expansion of that, which is why I think for the first stage of some of these things, we would like to see the greater development of these treatments are clearly defined as worthwhile. You could give A or B, but in other situations it's not clear whether you should give A or B, and that becomes in the guidelines also therefore listed as standard of care. I'll just give you that example that I mentioned. There's a trial at the moment in Canada that we're looking at possibly joining, which is looking at two forms of factor Xa inhibitor anticoagulant therapy for patients with DVT, pulmonary embolus. Both are in standard of care, and that's being done at the moment as a clinical trial, which has gone to an ethics committee. These are all the risks and benefits of therapy. To me, they're both listed on with the TGA and with PBAC as treatments that are funded for that indication. To me, that if you wanted to give one of those, rather than having to join this trial because it's there, that's great. But if it wasn't there, you should be able to go ahead and do it and just list that as one of the treatments that you're considering under this protocol.
Yes.
Yeah, John.
So you also.
No, go ahead, Stephane. Sorry, you go ahead.
Yeah. You said in your presentation that in a new paradigm, you would not necessarily go for a fixed sample size or even a non-treatment effect. So you would like to use existing information. So you are making the case for using this for using Bayesian methods for that sort of trial. Is this what you had in mind? Because you may get some information about different standards of care from what people are using or will be interested in. Plus, you have some other information about the treatments for a particular group of patients who don't really know exactly the effect. You could use that information to design your trial or define some particular test or comparison you want to do. Would you do it the Bayesian way? Because it would make a lot of sense to me.
I think the approach of adding the evidence to it, to the cumulative, like a cumulative meta-analysis to the existing evidence for those things, mirrors a Bayesian philosophy about how you're using things. So it's not that you. And the idea of that evidence, rather than saying, well, we've now got enough evidence, we should stop and recommend that people stop randomizing to that, That's not the philosophy here. The philosophy is to get that evidence back out to the clinician. So we're now saying that for this particular condition, this is now the latest evidence. If they say, well, look, based on that evidence, that's enough now that I should stop giving treatment X, or if people updating the guidelines say that treatment's no longer available, they do so. But if for the next patient in front of me, I still don't think that for them the evidence applies sufficiently, I could still go ahead and randomise. So I think it is more of a Bayesian approach rather than, there's no fixed sample sizes or stopping rules in that sense for this approach.
Yeah, for me it makes a lot of sense to do it the Bayesian way. It's natural to do it that way, so thanks.
Yes, I concur with Stephane. I mean, I was thinking of the frequentist approach, just I don't even know how you'd structure it. You'd have to model a whole system in advance. It would be virtually impossible.
Not well defined. You have several comparisons to different standards of care. You may have pairwise comparisons, depends what you want to do, and this can be really done easily in the Bayesian setting and frequentist setting. It's difficult.
So can I come back to the issue of dropping particular treatment arms or introducing new ones, and what didn't quite catch what you said there, John, about the basis for when you discontinue a certain arm or when you bring in a new one based on, well, the new one's possibly easier based on external evidence, but for discontinuing an arm, you're saying that they would use information from outside the trial as well, rather than just showing inferiority within the trial.
So the radical idea about this is that the clinical trial protocol, in terms of what questions you're looking at, is in the mind of the clinician and the patient, based on them being informed for their patient what the evidence is. So you're dropping an arm, as you put it, when there is sufficient evidence that people no longer regard that as an acceptable standard of care, because there's now sufficient evidence that the other treatment that we're being compared with are now the standard of care. So the mechanism for dropping the arm is not that we've officially in SOCRATES said this treatment's no longer an option. What we've done is tried to ensure that the evidence database for that has been updated and the guidelines have been updated, and people no longer use that as a standard of care in practice. That's the way a treatment would no longer be used.
Is there going to be a problem with time lag in getting that information to update guidelines, and by the time that's done, you may have had, a couple of hundred patients in the interim?
I don't think so. I think the time lag is exactly the same for any clinical trial. And also the issue is, if I just give you a couple of examples, we did a trial in tamoxifen prevention in breast cancer, where a study in the United States showed this reduced the incidence of breast cancer. So people said the trial stopped early and said, we've got our result. The trial we were doing in Australia and the UK said, well, look, we might be reducing the incidence of breast cancer, but what happens long term? Does this translate into other things? So our trial continued for a number of years after that. And it's a similar situation here. You're looking at this Socrates thing, adding to the global evidence for particular outcomes, and that if that evidence, so if we'd taken the evidence from the trial that was happening in the US and said, they've answered the question, we should also stop our trial in Australia. So it's using that mechanism rather than just looking at the evidence within Socrates as to whether we've answered that question.
Mm. Yeah, in the remap, in the community-acquired pneumonia trial, there's also the added element of response-adaptive randomisation. There's a lot more done within the trial, using data from within the trial itself, than I think what you're proposing there. And I can guess there are pros and cons for that, where treatment arms that seem to be doing better are allocated higher response probability, allocation probability, and conversely those that are doing worse get less probability, and then eventually to the point where they're eliminated altogether. And I've just got, sorry, may I ask a question to Stephane about remap cap, about when an arm is dropped, is external evidence that may have accumulated to the trial used in the decision about dropping an arm?
That was a question for you, Stephane.
Yeah, so Stephane?
I mean, I don't know. I mean, I couldn't hear everything you said. On sometimes the connection drops. But
Andrew, I think just one.
Yeah, we're having trouble hearing you.
Well, I didn't get the question. I'm sorry.
Can we, maybe we can drop my video.
Yeah, the connection is getting worse. I'm sorry.
Can I make some comment about that, though, in relation to the question?
Yeah, sure. Yes.
Study. Look, I think adaptive designs with modifying things with response, etc., are important ones, and I think they have great efficiencies, but I think they're mainly for evaluating potential new therapies rather than trying to best define what are currently regarded as standard care options. So I think the approach here will be looking at contributing to the total evidence base for which you already have evidence that it's a standard care option, rather than doing something just within the context of the trial. I think there's a great role for adaptive designs, particularly in the more novel therapy, where you're screening a number of approaches.
Yeah, I think we're in absolute agreement for the general concept of embedding trials in routine care that we've talked about here, and then these are the designs of the future that can be almost sort of self-learning. So what you've talked about, external evidence coming about, that down the line we could automate the transfer of data or the availability of data from the SOCRATES trial into whatever sort of global evidence base. Guidelines can be updated more quickly and then fed back into the system.
That's why I think some of the stuff is good to think 30, 50, 100 years' time as well, because some of this, if you say, oh, well, that's not going to happen in the next 5, 10 years, I'd agree completely. All I think we can demonstrate in the short term is that for ethical and other reasons, that this strategy can be adopted, which is a radical departure from what ethics committees currently do for trials, and which is limited, I think, to these standard care approaches. But in the future, if we can then build on that and make it as a more routine and seamless way that we are continuing updating our evidence bases electronically in the clinics, etc., then I think that's what the world will often look like for some of these things, and making the best use of randomised evidence.
And then you're just going ahead too, when we've got sort of genomic profiles on every patient, and then the standard of care treatment could depend on their genomic profiles. I mean, the options are tremendous for the future.
Exactly.
Yeah. Do we have any questions here? Oh, I see. She's giving me some technical advice. Oh, yeah. Stephane, any final comments before we close off we finish up this session?
Got into technical issues. I couldn't hear most of the conversation for a while, and it came back. It's a bit annoying. So no. I mean, I don't have any further comment. I've missed most of it, and I'm sorry about that.
Yeah, and apologies to everyone if the audio had been cutting in or out. So thank you very much, John, for giving us an insight into the future. I've got no doubt personally that's where trials absolutely need to be going for evaluation of routine interventions anyway. And thank you to both speakers, David and to John, and thank you to our discussants as well. And thank you to everyone for joining in. And when you log out of the system, please don't forget to give a score to the presentations. And a reminder that in 2 weeks' time, so that's on May the 15th, again at 10:30 a.m., we'll have our next novel designs session with 2 speakers talking about a different set of novel designs. But thank you again very much to John and David for these really stimulating presentations, and really an idea how clinical trials can look in the current, as with David, and really looking to the future, as John is doing. Okay, so thank you, everyone, and hope to see you all in 2 weeks' time. Okay,
Hey,
bye.
thank you.